All right, we're going to do a little demonstration of surface sterilization for leaves for the purposes of quantifying leaf phylosphere biodiversity. Our objective here is to record the diversity of fungi that are associated with the leaves of individual species, both in the canopy and in the forest floor. Everyone has been assigned a species, and your objective in sampling is to go out, find 5 individuals of that tree or shrub, and from each of those individuals collect 5 leaves in the canopy and 5 leaves in the forest floor. So 5 trees or shrubs, and then 5 canopy leaves and 5 forest floor leaves. Our objective is to analyze and understand the microbial communities, specifically the fungal communities that are associated with these 2 very different environments associated with these plants. All right, once you've done that, that field portion of your sampling, we're going to need to surface sterilize your leaves. This step is done in order to get all things that just happen to be on the surface of your leaves off. There are many microbes, lots of bacteria that are on the surface of the leaves, and they're there by chance. They're not associated with the biology of the plant. They're not the saprotropes, mutualists, or pathogens that we're actually interested in quantifying. We're going to limit our inferences. Sometimes they are, even though they're on the surfaces, but we're going to limit our inferences to things that are actually growing within the leaves, within the interstomatal space, or in the cells of the plants themselves. So the way that we do this is we surface sterilize in 2 steps. First is a 10% bleach solution. I've got a beaker of water here, and then I've got a beaker of ethanol as well. And what I do is I'm going to put my leaves, just imagine me having 5 leaves from the canopy of this sumac here, and I'm going to put this into this bleach solution. 10% bleach, household bleach, will damage your clothes, so be careful with it, but it is generally not hazardous to you, not any more than any other household chemicals. I'm going to soak this in here for 5 minutes, or excuse me, 3 minutes, so just imagine that that happened. I take it out, quickly dip it in the water, and then I'm going to put it into the ethanol for an additional 30 seconds. So this is for demonstration only, so just imagine that I have done that. I'm then going to put it in the water again to get the extra ethanol off, and then I'm going to put it down here on my paper towel so that I can process it. While I'm doing this, to be efficient, I take my next sample, the forest floor leaves that are associated with this individual tree, and I'm going to put them into my surface sterilization solution. That makes everything more efficient for me. Now, to do this work, you need a set of scissors. They can be fine scissors like this that are you would have for a first aid kit, or they can be and a pair of tweezers that likewise would be something that maybe is appropriate for a first aid kit. And you need to get these things sterilized. So you put them in your alcohol, whether it's your ethanol or isopropyl. And what I'm going to do is I'm going to take these, and I'm just going to dump a little ethanol onto these, onto this paper towel, and then I'm going to wipe my instruments off. You need to sterilize your tools between every sample, and in particular, between every, when you're switching from the canopy to the forest floor. Basically, sanitize your tools whenever you switch among trees or between strata. Okay? So what I'm aiming to do here is get about hole punch size samples from my set of leaves. And again, you got to imagine me having 5 leaves for this individual tree. And I'm just going to cut up, I would just cut up, say, 15 bits, working my way across all of the leaves that I sampled from the individual trees. I'm looking for things, it's nice to have them square, about 3 by 3 millimeters. That's about the size of a hole punch. you can actually, excuse me, I'm sorry, I'm working here without this visible. You can actually use a hole punch if you like. You can sterilize a hole punch the same way that you would scissors. So anyway, I've got these here. I've got my set of little things here, and I'm going to put them into my plates. I've got a plate. Remember, mark your plates on the bottom because the lid will rotate, and it will you'll lose track of what it is that you are what you have sampled. Here is a rather poor division of the media. I did a bad job, and if I was doing this for real, I would go back and I would erase this using ethanol or isopropyl, and I would remark it. But for the purposes of this video, I've just got one thing here. I've got one sample, tree 1. I've got it marked on the bottom of the plate. I've got my sterilized little leaf bits, and what I'm going to do now is I'm going to put them into their appropriate place on the plate. Now, here is good technique. I've got my plate like this, and I've got my left hand sitting on top of the plate like so. Excuse me. Got my left hand on top of the plate, and the reason for that is I'm going to try to open this plate for the shortest period of time possible. I am going to only open it long enough to actually get this little leaf bit in here. I'm going to take this leaf and I'm going to kind of saw down into the media so that it's standing straight up or it's in at an angle. So do that, and that's it. And I can work through all of my samples here for this particular sample, this set of leaves. I can again imagine it, so I sampled from 5 leaves. I don't have to surface sterilize my tool between every single one of these, especially if I'm moving pretty fast. If you when in doubt, if you have any concerns, if you drop your tools or something like that, then definitely surface sterilize in between. But really what you end up with is you should have a set of bits, and I should also mention you want them, use all the space in the plate. That way it will help you once things start to grow out. It will be easier to distinguish what you're looking at. Okay, so let's take a quick look here. That's my samples right there. You can kind of see them. They're in the plate. This is not actually going to be grown out, so I'm going to open them, and you can maybe see that they are half sticking up out of the media, or there's parts of them that are sticking up out of the media, and then the rest of them are in the media. You're only going to get stuff out of the material that is actually in the media. The portion of the leaf that you've cut up and is in contact with the media, that's where these fungi are going to grow out. For the most part, or the ones that are going to grow out in the time period of this particular study that we're going to do. So get the material into the media. Don't put it on top of the media, saw into the media and get it in there. The other thing that can go wrong here is if you cut your pieces of leaf too big. So, 2 by 2, 3 by 3 square millimeters of material is all you need, or about a whole punch. Enjoy. Really quick, fun things grow out, and I think you'll be surprised by them, and they're pretty cool to look at as well.